So in this chapter, basically what we'll be talking about is we'll be looking at genes and how they work. And genes and how they work, we'll be looking at things like cell division. So we'll look at cell division, that is mitosis and meiosis. And we'll also be looking at things like monohybrid inheritance and the difference between like sexual and asexual reproduction in relation to how DNA works. So let's start with mitosis. Now, cells divide, right? And if you go back to when we were discussing reproduction, we talked about how the single cell, which is the zygote, divides and divides and divides. And as the number of cells increase, the size of the organism increases. So when the sperm cell meets the egg and a single cell is created out of that, that one cell is what will grow into an entirely new individual, a new human being, right? So from that one single cell, you have division and division, and it continues to divide and multiply. So as it's multiplying, the number of cells will increase, right? And that is where you have cell division. And there are two types of cell division that we're going to look at. One is called mitosis, which is mitosis, and the other is called meiosis, meiosis, right? So let's start by looking at mitosis. So when you think of the human cell, we know that there are chromosomes in the human cell, right? Humans have 46 chromosomes, which is what I talked about a lot when we were looking at reproduction. So humans have 46 cells, 46 chromosomes in each of their cell, with the exception of one type of cell, but we wouldn't talk about that now. So your skin cells, your eye cells, hair cells, nail cells, heart cells, liver cells, all of those cells contain 46 chromosomes, right? 46. Good. When your skin, let's look at this for example. When you're younger, or even when you're older, you get an injury. So it's maybe a scratch, a cut, a dig, something like that. After a while, it heals. And when it heals, new skin grows back. What is happening there is cell division. So whenever you get an injury, the cells in your skin divide and new cells are born, new cells are formed. And so that is how injuries manage to heal, because the cells are dividing and replacing the old cells that have been killed, or those old cells that have been damaged when you receive that injury. New cells are being made to replace the old cells. And that is how you're able to heal, right? A child, when a child grows, they grow in height. So they get taller. How is the skin working to fit the body of that child? So if you look at a baby, you may think, oh, well, the baby skin grows. Because how does a baby go from being like maybe small as the length of your arm to being really tall? Where is all of this extra skin coming from? Because if they're increasing in size, then the skin must also increase to be able to cover up the child, right? So where is all of this skin coming from? It's not that the skin is stretching. The skin is actually growing along with the child. So as the bones of the child grow and the child gets taller, the skin is also growing. So how does the skin grow? Well, the skin is made of cells. And when these cells divide, that is how it grows, right? So you think about it, let's say for instance, let's think about something that maybe it multiplies. Let's think about mosquitoes, right? So you start off with maybe 1, 2 mosquitoes, and the mosquito multiplies, so it makes young ones, and the young ones will eventually grow and make young ones, and those will eventually grow. So at the end of maybe, let's say, a month, you will have more mosquitoes than you started out with in the first place. So the amount of mosquitoes that you started out with has increased drastically. The same thing happened with your cells, right? So when your cells divide, they increase because they're multiplying. And that is how the skin is able to grow. If you think about it from the perspective of a child growing or your skin healing from an injury. So now we're at mitosis. Let me use dough as an example. I think like that's the easiest example I could use. When you are making, let's say making roti. I don't know if y'all know for making roti or you ever see people making roti, but let's deal with roti, right? So you're making roti. At first, it starts as one dough, right? So you mix up the dough, you mix up the flour and everything. So it starts off with one dough. And then you pinch that dough into smaller pieces and you roll it out and they become their individual dough, right? And then you bake the roti. Good. That is how your cells divide in the process of mitosis. First, you have one cell, and that one cell splits and becomes two cells. Each of the two cells will then split again and become 4 cells. So that is how, and mind you, when they split, they don't decrease in size, right? So it's not like half and half when it splits. When it splits, it becomes 2 individual cells, right? That is mitosis. When one cell splits and become 2 cells, and then it splits again and it continues to split and divide, and that's how it multiplies, right? That's how the cells multiply. So let us look at mitosis now specifically. So you have a cell. In the cell, there is the nucleus of the cell, right? Which is relatively in the middle of the cell. You have the nucleus. In the nucleus of the cell, there is where your 46 chromosomes are located, right? Your 46 chromosomes are located there in the nucleus. 23 of that 46, which is half, you got from your mother, and the other 23 you got from your father. That is how you got 46. 23, 23. The 23 comes together, and that's how you get 46, right? When the 23 comes together, they line up next to each other. So kind of like when you're in school and you gotta line up, they line up next to each other, right? So I want you to think about when you're lining up in school, you got boys in one line, girls in one line, right? And if you were to match the first girl with the first boy, the second girl with the second boy, you do that, right? And think about the first girl and the first boy holding hands then the second girl and the second boy holding hands, then the third girl and the third boy holding hands. So you do that all the way to 46. So the chromosomes are positioned in pairs in the nucleus of the cell, right? The chromosomes are located as pairs. So one from the mother and one from the father comes together and they make a pair. This happens with the second set of chromosomes, the third set, the fourth set, just like the children who are lining up. So that is how you have 46 chromosomes, and they are situated in 23 pairs. Okay, so in mitosis, when the cell divides, two identical cells are formed. Let me tell you what that means. You have one cell, and when that one cell splits into two, they become two individual cells, right? So it's not half and half of a cell and half of a cell. They become their own cells. So the first cell, we're starting off with 46 chromosomes, and when it splits, each cell will have 46 chromosomes, right? It doesn't split half and half. And this is what we're going to go through now. How is it that we start off with 46, but when it splits, you still have 46 in both? Just give me a minute. Let me pull up my notes. All right. So the reason why this happened is because the chromosomes are duplicated. So copies are made of the chromosomes. So when you go to the printer and you take a photo and you say, okay, I want 5 copies of this. The original copy is duplicated, right? And the 5 other copies that are made are identical to the original copy. So the same thing happens to the chromosomes. Before the cell actually splits into 2, the 46 chromosomes are duplicated so that you have a total of 96 two chromosomes in the cell. This is when it's going to divide, right? This is not all the time. This only happens when the cell is about to divide. So the chromosomes are copied, the 46 are copied, and you have a second copy inside of the cell, which is another 46. At that point, the cell will then split, and as it splits in half, you'll have 46 in one and 46 in the other one, right? At the end of mitosis, you have two daughter cells. We call them daughter cells because basically they came from the same parent, right? So we call them daughter cells, so that you'll know that the cells that we're talking about are the ones that were derived from the original cell. So you have two daughter cells. The second thing is that we call them clones because they are identical to the parent. So they're clones, right? There is no difference between them. It's like identical twins. They are identical. They have the same DNA. That's why they look the same, right? You and your brother and your sister, you look different from each other because you're all carrying different sets of DNA. So you look different, right? But in identical twins, they're carrying identical DNA. That's why they are identical. And so in the cell, because the cells are identical to each other, we call them clones. Thirdly, you say that at the end of mitosis, each daughter cell contains the diploid number of chromosomes. Diploid means double, right? So double would be double of 23, which would be 46. So it's another way of saying that after mitosis, each cell contains 46 chromosomes, or the diploid number of chromosome, diploid, diploid. You need to remember that word, right? Because that word will come up again. So let me go over the features of mitosis. Mitosis occurs in all the body cells except one specific cell, which I will talk about later. Secondly, at the end of mitosis, you have two daughter cells. Thirdly, the daughter cells are clones. The fourth feature is that at the end of mitosis, you have the diploid number of chromosomes, or 46 chromosomes in each cell. Good. So let's go to the division now. This cell division is divided into stages, and you use the acronym PMATI to remember the different stages of the cell division. PMATI. P is for prophase, M, metaphase, A, anaphase, T, telophase, and I, interphase. So let's start with prophase, which is P. In prophase, the cell is saying, hey, it's time to divide. Let's get prepared. So the cell starts to make preparations, right? The cell is making preparation to start its division. So that's in prophase. In metaphase, the chromosomes line up. So when I say they line up, this is what happens. The children who were holding hands in the line will loose off of each other, and instead of standing side by side, they will now stand one behind the other, right? So instead of two lines, you now have one line of students. Both boys and girls are lined up in one line. So that is what happens in metaphase. And remember, I'm using the children to mean chromosomes, right? So the chromosomes will unravel from each other, because remember, they're in pairs. They will unravel from each other and line up side by side along the center of the cell. So in the middle of the cell. Um, you know, when you're looking at a map of the earth, right? You see the equator, and the equator is a line drawn right through the middle of the line going the earth going from left to right. That's how the chromosomes line up, left to right in the middle of the cell, right? Along the middle of the cell, because they're getting ready to divide. Good. At this point, the chromosomes will then shorten and condense. So they are like becoming thicker, because remember, they will have to split. So just imagine now that each child that is lined up in the line, an identical copy of them will be made, right? So that happens in metaphase and anaphase, right? Now, when the duplications are made, so let's say each child in the line now has been cloned. So you now have two lines with identical children in the line, right? In telophase, the new set of clones will move to one side, and the previous line of children will move to the other side. Then is when the cell will split. When the cell splits, you'll have 46 chromosomes in one cell and 46 chromosomes in another cell, right? And then they will then regain their original position. So the boys will separate from the girls like how they were before. And this will now happen in both cells, right? The girls will separate from the boys, and then they will go back to holding hands. They will retake their former position. But now you have two cells, and in each of the cell, you have 46 chromosomes. So let me put this in the terms that you have to know. The cell prepares itself for division. The chromosomes will unravel themselves and line up side by side along the center of the cell. These chromosomes will condense, and then they will be duplicated. So identical copies are made. When that happens, 46 will be pulled to the top of the cell, so the northern pole of the cell. 46 will be pulled to the top, and 46 will be pulled to the bottom of the cell. So now you have 46 at the top, 46 at the bottom. Then is when the cell will split in the middle. Okay, that sounds like a song, right? Well, then in the middle. And when it splits, right? When it splits, each cell now will have 46 chromosomes, and the chromosomes will now regain their former position of being wrapped around each other in pairs. So you can understand why we call them clones, because the DNA has been duplicated. It is not new DNA that has been made. The same DNA that was in one cell has been copied and duplicated, and that is what is present in the other cell, right? So that is mitosis. In interphase, interphase is the resting state of the cell. Remember, the cell is not dividing all the time. So when the cell is not dividing and when it is doing its normal everyday job, that is what we call interphase, until it is ready to divide again, and then it will enter into prophase, metaphase, anaphase, telophase. Now we come to meiosis. Good. Remember I said before, mitosis occurs in all of the body cells except one specific cell. That one specific cell is the gamete or the sex cell. So in males, we refer to this as the sperm cell, and in females, we call it the ovum or the egg cell, right? The way these cells divide is different to how ordinary cells divide in your body. That's why this is a whole new process called meiosis now, right? Good. Can anybody tell me why it is different? Looking at what we did for reproduction, how many chromosomes are found in the sex cells?
Miss 46 chromosomes.
46 is found in the sex cells. All right, I want you to reason it out. If 46 chromosomes are found in the sperm cell and 46 chromosomes are found in the female egg cell, when it fuses, how many will you get in total? 92.
Wow.
Is that the normal number of chromosomes for a human, 92?
I don't think so.
It isn't. Humans only have 46, right? Humans only have 46, which means that the sex cells only carry half, which is 23. The other cells in your body that make up your organs, your skin, your eyes, all of those things, those cells contain 46 chromosomes, right? The only cell in your body that does not contain 46 chromosome would be the sex cells. In males, it would be the sperm cell. In females, the egg cell. The reason why this is because the sperm was meant to be fused with the egg, right? So you understand why it is impossible for each to have 46, because when it fuses, you get 92, which is impossible. That's not a human anymore, right? They're gonna look like something else. They're gonna look like an alien or something. It wouldn't look human. So in order for this to be normal, the male sex cell, which is the sperm, must only carry 23, and the female sex cell must also carry 23. So when they both fuse, it will make the 46, right? That is why they're the only cell in the body that only carries 23. Because,
Miss, the last thing I hear you say is Doug, look, the internet just gonna come so it bunks me off.
okay, but I'm recording it, so you'll be able to listen back.
Okay.
Right, so as I was saying, only the male and the female sex cells contain 23 chromosomes because they were meant to be fused together. And when they're fused together, that is when you'll get the 46. That is why they don't have 46 each, they only have 23. So here's what I'll do. I will find, hold on. All right, let me show you this video. All right. Good. So I'm going to play this video now.
When the cell splits apart. What is mitosis? Eventually, cells need to duplicate. There are two main methods of replication, mitosis and meiosis. This tutorial will talk about mitosis. The big idea to remember is that mitosis is the simple duplication of a cell and all its parts. It duplicates its DNA, and the two new cells, daughter cells, have the same pieces and genetic code. Two identical copies come from one original. Beyond the idea that two identical cells are created, there are certain steps in the process. Before a cell divides, it is in resting phase. The cell in resting phase is not actually resting, since it has to maintain its reservoir before it undergoes division. This phase is called interphase. It prepares the cell for division, i.e. protein synthesis, DNA duplication, and enzymes. After this phase comes mitosis, which is further divided into karyokinesis, division of nucleus, and cytokinesis, division of cytoplasm. Karyokinesis involves the four basic steps of mitosis that are prophase, metaphase, anaphase, telophase. Here we start with prophase. The chromosomes start coiling and become condensed. Nuclear envelope disintegrates. Gene transcription ceases during prophase and does not resume until late anaphase to early G1 phase. The nucleolus also disappears during early prophase. Centrosome gives rise to mitotic apparatus, i.e. generation of spindle tubules. Metaphase. Now all the pieces are aligning themselves for the big split. The DNA lines up along a central axis, and the DNA, chromatin, has now condensed into chromosomes. Two strands of chromosome are connected at the center of the centromere. The tubules connect to the centromere, not the DNA. Anaphase. Here we go. The separation begins. Half of the chromosomes are pulled to one side of the cell. Half go the other way. When the chromosomes get to the side of the cell, it's time to move on to telophase. Telophase. Now the division is finishing up. This is the time when the nuclear envelope begins to synthesize again along the separated chromosomes. Cytokinesis pinches the cell membrane in the middle. Cell membrane closes in and splits the cell into two. Now we have two separate cells, each with half of the original DNA. Up till now, we have discussed what is mitosis and its different phases. Now it's important to know about significance of mitosis. Significance. It is an equational division through which identical daughter cells are produced, having the same amount and type of genetic constitution as that of the parent cell. It is responsible for growth and development of multicellular organisms from a single-celled zygote. The number of chromosomes remains the same in all cells produced by this division. Thus, the daughter cells retain the same character as those of the parent cell. It helps the cell in maintaining proper size. Mitosis helps in restoring wear and tear in body tissues, replacement of damaged or lost part, healing of wounds, and regeneration of detached parts, as in the tail of lizards. It is a method of multiplication in unicellular organisms. If mitosis remains unchecked, it may result in uncontrolled growth of cells, leading to cancer or tumour. The next important phase of cell cycle is meiosis. Have you ever wondered why you are different from your siblings? The answer lies within the amazing process of meiosis. What is meiosis? Meiosis is a process where a single cell divides twice to produce 4 cells containing half the original amount of genetic information. These cells are our sex cells, sperm in males, eggs in females. During meiosis, one cell divides twice to form 4 daughter cells. These 4 daughter cells only have half the number of chromosomes than the parent cell. They are haploid. Meiosis takes place in germ or sex cells called gametes, eggs in females and sperm in males. Similar to mitosis, cells also pass through the interphase, G1, S, and G2 phase before they enter meiosis. Here are the distinct phases of meiosis. Interphase, meiosis 1, cytokinesis 1, meiosis 2, cytokinesis 2. First at interphase. In interphase, DNA is copied, resulting in 2 identical full sets of chromosomes to prepare for division. Outside of the nucleus are 2 centrosomes, each containing a pair of centrioles. These structures are critical for the process of cell division. In next step at meiosis 1 takes place in following steps. Prophase 1. Prophase 1 is typically the longest phase of meiosis. During prophase 1, homologous chromosomes pair and exchange DNA, homologous recombination. The new combinations of DNA created during crossover are a significant source of genetic variation and result in new combinations of alleles, which may be beneficial. The paired and replicated chromosomes are called bivalents or tetrads, which have 2 chromosomes and 4 chromatids, with 1 chromosome coming from each parent. The process of pairing the homologous chromosomes is called synapsis. At this stage, non-sister chromatids may cross over at points called chiasmata, plural, singular chiasma. Prophase 1 has historically been divided into a series of substages, which are named according to the appearance of chromosomes. Leptotene. In this stage of prophase 1, individual chromosomes, each consisting of 2 sister chromatids, become individualized to form visible strands within the nucleus. Leptotene is of very short duration, and it's when the progressive condensation and coiling of chromosome fibers takes place. Zygotene. Chromosomes approximately line up with each other into homologous chromosome pairs through synaptonemal complex. The paired chromosomes are called bivalent or tetrad chromosomes. Packetine. This is the stage when homologous recombination, including chromosomal crossover, crossing over, occurs. Non-sister chromatids of homologous chromosomes may exchange segments over regions of homology. At the sites where exchange happens, chiasmata form. Diplotin. The synaptonemal complex degrades and homologous chromosomes separate from one another a little. However, the homologous chromosomes of each bivalent remain tightly bound at chiasmata. The regions where crossing over occurred, the chiasmata remain on the chromosomes until they are at the transition to anaphase 1. Diakinesis. It closely resembles prometaphase of mitosis. The nucleoli disappear. The nuclear membrane disintegrates into vesicles, and the meiotic spindle begins to form. Synchronous process. During these stages, two centrosomes, containing a pair of centrioles in animal cells, migrate to the two poles of the cell. The microtubules invade the nuclear region after the nuclear envelope disintegrates, attaching to the chromosomes at the kinetochore. The kinetochore functions as a motor, pulling the chromosomes along the attached microtubule. Microtubules that attach to the kinetochores are known as kinetochore microtubules. Other microtubules will interact with microtubules from the opposite centrosome. These are called non-kinetochore microtubules or polar microtubules. Metaphase 1. Homologous pairs move together along the metaphase plate. The paired homologous chromosomes align along an equatorial plane that bisects the spindle. Anaphase 1. Kinetochore microtubules shorten, pulling homologous chromosomes, which consist of a pair of sister chromatids, to opposite poles. Non-kinetochore microtubules lengthen, pushing the centrosome further apart. The cell elongates in preparation for division down the center. Telophase 1. The first meiotic division effectively ends when the chromosomes arrive at the poles. Each daughter cell now has half the number of chromosomes, but each chromosome consists of a pair of chromatids. The microtubules that make up the spindle network disappear, and a new nuclear membrane surrounds each haploid set. The chromosomes uncoil back into chromatin. Cytokinesis, the pinching of the cell membrane in animal cells or the formation of the cell wall in plant cells occurs, completing the creation of two daughter cells. Sister chromatids remain attached during telophase 1. Cells may enter a period of rest known as interkinesis or interphase 2. No DNA replication occurs during this stage. Meiosis II. Meiosis II is the second meiotic division and usually involves equational segregation or separation of sister chromatids. Mechanically, the process is similar to mitosis, though its genetic results are fundamentally different. The end result of meiosis II is production of 4 haploid cells, N chromosomes, 23 in humans. The 4 main steps of meiosis II are prophase II, metaphase 2, anaphase 2, and telophase 2. In prophase 2, we see the disappearance of the nucleoli and the nuclear envelope again, as well as the shortening and thickening of the chromatids. Centrosomes move to the polar regions and arrange spindle fibers for the second meiotic division. In metaphase 2, the centromeres contain 2 kinetochores that attach to spindle fibers from the centrosomes at opposite poles. This is followed by anaphase 2, in which the remaining centromeric cohesion is cleaved, allowing the sister chromatids to segregate. The sister chromatids by convention are now called sister chromosomes as they move toward opposing poles. The process ends with telophase 2, which is similar to telophase 1 and is marked by the disassembly of the spindle and decondensation and lengthening of the chromosomes. Nuclear envelopes reform and cleavage or cell plate formation eventually produces a total of 4 daughter cells, each with a haploid set of chromosomes. Meiosis is now complete and ends up with 4 new daughter cells.
I wanted to learn somewhere where I could go anywhere else in the world. I'm still able to treat other Korean people in New York City. I love my job.
We're the #1 provider of doctors in first-year U.S. residencies. St. George's University School of Medicine. Are you in?
All right, so here's what I'll do. I'll stop there for today because I am